In 1856, Gregor Mendel gave us our first look into the importance of genes. Today, we're enticed by exciting new treatments for diseases, genetics, and your health tonight. On Call with the Prairie Doc. Good evening and welcome to On Call with the Prairie Doc. Tonight, our eyes turn to the rapidly evolving medical field of genetics. Over the centuries, scientists have developed an understanding of the genetic code entwined in a double helix within every cell. It's the template that makes each of us look and act like a combination of our mother and our father. Now, the human genetic jigsaw puzzle has been put together, and there are wonderful medical consequences for such knowledge. More about this tonight. But first, let's take a look at this week's Prairie Doc quiz question. Who established the concept that evolution occurs as a result of natural selection and survival of the fittest? A. Gregor Mendel, B. Charles Darwin, or C. Jean-Baptiste Lamarck? We'll give the answer to that question at the end of the show. Well, joining us tonight is Dr. Cassie Hajek, Chief of the Division of Genetics Medicine at the USD Sanford School of Medicine. And also with us is Dr. Erick, Scientific Director with the Avera Institute for Human Genetics. Thank you both for joining us.
Thank you.
Thanks for having us.
So, Cassie, tell us a little bit about yourself. I mean, you didn't start off as the chief of genetics. I mean, what did you train, and what did you train as?
Yeah, so I trained as an internist. I
Like me.
Like you, yes. And I love internal medicine. I love primary care, preventative health. But that was also one of the things that sort of drove an interest in genetics.
Right.
Started to kind of think about things that maybe present like a common disease but aren't necessarily a common disease, and as I went through genetics training at UCLA, I found that there are a handful of things that are like that, and so it just piqued my interest. And then I also have an interest in common disease genetics, which is an evolving field but not quite ready for prime time, I would say. So I went back for training, and here I am practicing adult genetics and working with the School of Medicine and our residency program to help increase education in that area.
It's fascinating, and it's going to evolve right before your very eyes and blossom and flower as you develop in your field. I'm thrilled for you.
Yeah, it's been really fun. I mean, even in the short 2 years of my training,
everything's changed.
Everything's changed. When I got there, we were looking at exomes, and when I left, they were starting whole genome on a very small subset of patients. But to see like such a rapid clinical transition was fascinating, and it continues to be.
Right. Well, and then Erick, you are a PhD in molecular genetics.
Right.
You're originally from South Dakota?
Yep, born and bred, raised in Aberdeen, South Dakota.
Oh, you're an Aberdonian.
Yeah, Aberdeen. Yeah. And as a young boy, I always had a fascination with math, science. As I started to get older, I had a keen eye towards healthcare, and really wanted to utilize my math and science knowledge to help people. So one of the first things I did was I got a nursing degree at South Dakota State University.
Oh, you're an SDSU grad.
I've been throughout the regential system in South Dakota.
And then you went to USD, so you've been to both.
I went to Northern State.
Oh, and
I got my bachelor's degree in biology, and then actually went to the University of South Dakota and got my master's degree in microbial genetics, and then went to SDSU and got my registered nursing degree. And worked at Avera McKinnon as a registered nurse for a little over a year. And then really, at that time, really wanted to help patients, help the people I worked with using research. So I got a job at the Avera Institute for Human Genetics as it was inaugurated in 2006. And then during that time, I went to USD and got my PhD in molecular genetics. And then have been working there since and trying to really translate what we do at the bench to the bedside and helping our patients in healthcare.
Well, that's the rub, isn't it? We've got a lot of bench stuff, but what can we do to make it clinically helpful? I think the whole field of immunogenetics and turning on the immune system is the hottest area. We talked a little earlier about cancer therapy with finding a particular part of a cancer cell and then giving a vaccine against it. Could you explain that a little bit?
Yeah, well, cancer is a disease of the genome. In order for a normal cell to turn into a cancer cell, something has to happen to the DNA. There has to be some sort of mutation, some sort of trigger that causes that DNA to mutate and cause that cell to grow uncontrollably. If we can identify what that mutation is and how that affects the proteins on the surface of the cell, we can utilize our immune system, our T cells, and charge them up against that certain protein, that faulty protein, and utilize them as almost a vaccine to target those faulty cells and to remove them from the tumor, almost as it would remove a bacteria or a virus.
And that's actually happening. I mean, that's kind of theoretical stuff, isn't it? I mean,
It's theoretical, but it's being really put into practice. People are starting to utilize this technology in clinical trials to really see what the efficacy of this is, to see if there's any side effects related to this type of therapy. And hopefully in the future, this will be part of medical practice and it'll be FDA approved to utilize some of these therapies to help patients that have cancer.
But right now it's in the research level.
Right now it's in the research level,
yes. Cassie, any experience that you have that's similar? Because we have two systems. We have the Avera system, we have the Sanford system.
Right, and my experience is more in the clinical realm for cancer risk prediction. So,
right.
For example, from what we know now, about 5 to 10% of cancers are probably related to a genetic predisposition, meaning we can do a test and there may be a genetic mutation that has led to the cancer. For example, like BRCA1 and 2.
Breast cancer.
Breast cancer predisposition, breast and ovarian cancer predisposition.
Right. That's a BRCA1 and 2.
Correct. Yeah. And so we are able to do that, but it's not for everybody, right? It's for people who maybe there are red flags, like multiple family members with the early onset of the same type of cancer, or family members that have multiple primary types of cancers. In those cases, that kind of puts up a red flag. Maybe there is a genetic condition here, and there are genetic tests that we can do to help identify that.
But not everybody can. I mean, how expensive is that test?
It is, well, and that's an interesting thing. Those costs have been dropping significantly. So at this point, you can actually get an out-of-pocket test panel for as low as $250 because of the technology has advanced so rapidly, and we're learning so much. And then in some cases, insurance covers it. So it really depends on who your insurer is and
it's probably not appropriate to get it tested if there's just a general kind of a small, counseling is the important thing, right?
Exactly. It's definitely not appropriate for everyone. Like I said, there are certain situations where if the family history
very
is
strong
indicative,
for ovarian and breast cancer.
Yeah, or even just breast cancer. I mean, sometimes, and this is what we're learning, these things don't always present the same in every family. Some families will be more breast heavy, some will be more ovarian cancer. It's just, it really depends. But when you do have those earlier onset cancers, that's when you start to be worried about a genetic predisposition.
Right.
And then once you know that you have a risk for that, then you can do increased screenings.
Yep.
You can,
screen the other family
screen other family
maybe have
members,
mammograms
but also,
earlier.
Yeah, and a lot of these genetic conditions have additional cancers that people are at risk for. For example, breast cancer, the BRCA mutations, you may see an increased risk for male breast cancer. You may see an increased risk for male breast cancer, melanoma, prostate cancer, and so then in these individuals, you would really, sometimes the men get forgotten in these cases, and you really want to make sure that the family knows that there are other cancers that are associated with these genetic mutations that you want to screen for. And sometimes the most powerful thing is if you find a mutation in a family member, testing other family members, and if they're negative, then they're relieved from that additional screening, and they can be screened as from a population risk.
Right. So screening for cancer is one thing. Treating cancer is another. And counseling is one other thing. What about children with unusual characteristics that it would make a person want to do testing for that, for example?
So that's a broad category. I mean, we started with cancer. There are
other things to test
pediatric cancers, but other things to test for. There are several thousand rare conditions that all have a various phenotype or physical features associated with them. Behavioral changes. Some of the autism spectrum disorders are now we're starting to see a genetic predisposition to some of those, although it's a small percentage. But it's a reasonable thing to think about when you have someone who has a real diagnosis of autism to send them for a genetics referral.
And we can check for genetics on that. That's very interesting to me. We're going to talk about pharmacogenetics, but when we come back. Genetic counselors used to be a rarity, but as we learn more about genetics, the need for genetic counselors has grown.
So when I tell somebody I'm a genetic counselor, I get tons of questions. Do I help pick gender of babies? Do I help design human beings? No. I like to describe my job as a mix between genetics education and helping people understand what's going on with them genetically, if they have a certain condition or if there's something going on in the family, trying to help them understand that. And then also the counseling piece, helping a family deal with whatever condition's going on, manage it, tell family members, get resources. So kind of both that education and counseling pieces. I think our first genetic counselors were probably in the mid to late 80s, and there are only about 32 programs to train genetic counselors in the country, and it's numbers that are growing. We are up to about 4,000 genetic counselors now in the country, but the need is growing as we learn more about genetics, learn more about conditions that we previously weren't aware of. So a genetic test is a blood test. We have DNA in our blood cells, and we send that blood off to a lab that can isolate the DNA and then check to see is there any problems, or I like to call them spelling errors, within that gene that we're curious about. I work in adult genetics, so I see a lot of family histories of breast cancer, colon cancer, examples being Lynch syndrome or the BRCA gene. We see a variety of different connective tissue disorders and heart conditions like Marfan syndrome. Genetic counseling helps a family first understand their family history. We take a really thorough, we call it a pedigree, it's kind of like a family tree to help understand what do different family members have, how old did family members live, what types of conditions are running in the family. So that's the first place I start with patients I meet with, is just gathering that information and encouraging them to reach out to their families to gather that information. And then helping interpret that and explaining what it means to them, what the risks are to them, what the recurrence risks are, where do we go from here, how do we help manage that? And then is there any sort of testing that we should do to help confirm that genetically? What would you do if you did find out that you had this hereditary cancer syndrome? Would you think about having a prophylactic mastectomy like Angelina Jolie did? Would you think about removing your ovaries? Or some people opt for just increased surveillance. And so really figuring out a plan that feels right to the patient is important to talk about it up front. I think one of the misconceptions about genetic counseling is it's all about the genetic testing, that people come in for a blood draw, they come in for a test, and we do a lot more than that. Not every patient that meets with me needs a genetic test or is appropriate candidate for a genetic test. So really just basing our conversation off of the family history and what's going on, and helping provide either reassurance or direction and where to go with what's going on in the family.
So that was very interesting. Thank you so much, Megan, who happens to be a very dear family friend because her grandparents grew up in De Smet. Oh. and her grandfather was my hero, mentor, Dr. Bell, G. Robert Bell from D. Smith. So we're talking about genetic counseling, and there's different kinds. Erick, you mentioned to me simple versus Mendelian versus a complex.
Yeah, so when we talk about genetics, we can really break that down into two parts. There's the classical Mendelian genetics that we
Mendelian meaning after
Gregor
Mendel.
Mendel.
Yeah,
yep,
Gregor
yep.
Mendel.
And these types of disorders are one gene, one disorder. So a good example of that is cystic fibrosis. Another example is Huntington's disease or even sickle cell anemia.
That's one gene, one disorder.
You have the mutation in that gene, you're going to get that disorder 100%, no questions asked, no ifs, ands, ors, or buts about it. You'll get that disease, and we can test for that. Dr. Hajek was just talking about how we can take some DNA and look for that mutation in that gene, and we'll know if you have that disorder. That's in stark contrast to what we call complex genetics. And the classical Mendelian genetics are, those disorders are rare. They affect 1 in 100,000 people. The complex genetics, the second part, are much different. They encode for disorders that are common diseases.
Diabetes.
Heart disease.
Cancer.
Cancer. And that's not one gene, that's several genes, maybe 50 different genes, 100 different genes that each confer a really small risk to that disorder. But most importantly, there's also an environmental component. So you have an interaction between the genetics and the environment. A lot of times in genetics, we'll say the genetic factors will load the gun and the environment will then pull the trigger.
That's a nice way. It's not a nice.
Addiction is a good example, right? So you could have all the genes. You could have 10 genetic factors that put you at risk for nicotine addiction, right? But if you never take that first cigarette or you never are exposed to that environmental factor, you will not become addicted to that. So it's a classic gene and environment interaction.
Addiction. I was immediately into thinking smoking and cancer of the lung. And you've got the potential for cancer of the lung, but you've never smoked, so you don't get it. But you're talking about the addiction of smoking.
It can be cancer too. You can have genetic factors that put you at risk for cancer. Dr. Hajek was talking about the 5 or 10% of cancers that are a genetic predisposition. If you have that genetic predisposition, you still need the environmental factor to manifest the disease, whether for lung cancer, that's smoking, for melanoma, is that
too much sun.
Too much sun. The cancers are a gene and environment. It's a complex genetic disorder.
Just taking a family history is one of the things that I think as a general internist is important.
Oh yeah, yeah, and I think family history is still probably the best, predictor for various conditions. It's very helpful, especially in many different things. I always go to cancer because many people are affected by cancer. At least one person in their family is.
Yes.
Oftentimes. But knowing, being able to determine what kinds of cancer, who's affected, how old they are when they're affected, that really is very helpful when you're trying to determine whether or not there is a known single gene genetic predisposition, or if it's on the other end of the spectrum, like Dr. Ehli was explaining, more of a gene environment type of cause.
Right.
So
that's interesting. I think about those single genes, Mendelian situations with Huntington's chorea, and you look at, I took care of a woman way into her late 80s, into her 90s, and she had, I think, 4 siblings, and 3 of them died of Huntington's, and she didn't, and one other family member didn't.
yeah.
And she said it was like living with a gun to my head my whole life. But look, I've gone all these years and I didn't get it. Mm-hmm. So my children didn't get it either.
Mm-hmm. And that's a tough one too, when people are determining whether or not they want to get genetic testing for Huntington's, because
why no?
Why no? And I've experienced very different reactions, some of which I haven't expected when delivering results for Huntington's.
Really?
And probably the most shocking has been when we've given patients results that they're negative, and they come back with sort of
Disappointment.
kind of disappointment because they've lived their lives believing that they have a condition and maybe not had kids or maybe had taken a different career or done something completely differently because they've had this in their minds and their family and their really real life experience that this is what I'm going to have, and then they don't.
And it's but I would be overjoyed not to have Huntington's chorea. I mean, they've watched this.
Yes.
It's the worst. It's pretty bad.
It's pretty bad, and they've seen the worst of it. And my response is the same as yours. I thought to myself, gosh, why would you be anything but? But then you understand, if you've really planned your life thinking you have this one particular condition and you're going to die young and you're going to have behavioral issues and you're not going to be able to function.
Why go to college? They didn't go to college. They didn't do these things. They didn't have family.
So it's a very unique counseling situation. It's very difficult.
Well, and as Megan said, a lot of what she does is just communication and support. It's not really so much, here's your chances and so on and so forth. It's more, it's a counseling.
Oh, yeah, it's a counseling, and all of those things are important, all of the recurrence risks and additional risks and all of those things, but you're still telling somebody that potentially they have this health concern that is quite serious.
Yes.
Just helping them through the weight of that is a big part of that counseling.
Right.
And there's other implications too, and that's where the genetic counselor really comes in, because if you know your risk for Huntington's disease, that doesn't only affect you, that affects your children, that affects maybe your mother or your sibling. So even though you know your risk, maybe your sibling doesn't want to know their risk, or maybe your children don't want to know
the risk.
Or maybe they do,
wow.
So that type of genetic testing is not only for you, but it has implications for other people, and that's really where the genetic counselor can educate the patient and really explain to them the implications of knowing or not knowing and what they can do with that.
They're talking about knowing if a person's going to have Alzheimer's disease or not. And that's an interesting concept. Maybe we really don't want to know whether I'm going to have Alzheimer's disease or not. I mean, who would want to know that if there's nothing that you can do?
That's honestly the number one question I get. If I get testing, am I going to find out if I have Alzheimer's as an incidental finding? No, but because we don't have a treatment for it, and most people, that's they're really their number one fear. But right now, it's in a place where we don't really know. We don't know how to treat it. So, what does having that information do?
Not worth it. There's certain ignorance is bliss. There's a time when you just don't want to know. We talked about pharmacogenetics earlier before the show. Tell us a little bit about what we're doing with pharmacogenetics.
That's a really good question. Pharmacogenetics is a relatively new field of genetics. And really what it entails is the body's genes' response to medications and the drugs that we're prescribed and taking. Every medication we take has to be removed from the body. Most of these medications are excreted through the liver or the kidney, where certain genes that make proteins modify these drugs, and we release them from our body. But these genes come in different forms. So you may have a form of a gene that makes you a poor metabolizer for a medication. Or you may have a certain form of the gene in the liver that makes you an ultra-rapid metabolizer of a medication. Now you can understand the implications if you're a poor metabolizer and you get a standard dose of a medication from your physician. That medication is going to build up in your system, and you're going to be the individual that has the side effects, the adverse effects. You'll probably take
accidental overdose.
Maybe not accidental overdose, but you'll have the side effects. You may take your pills and want to throw them in the garbage because you just don't feel good when you take them.
You could be getting by with 1/4 of the dose.
Exactly, exactly. Some people have a form of the gene that makes them an ultra-rapid metabolizer, where you get the standard dose of the drug and your body clears it out of your system so quickly that you don't have any efficacy at all. You have no relief, for example, for pain. If you're an ultra-rapid metabolizer for a pain medicine, your pain won't be alleviated by that medication. So what we can do is take a DNA sample. We do it pretty painlessly through a blood draw, and we examine those genes that break down those medications. And we can, we have pharmacists that look at some of that information and can determine whether or not you're an ultra-rapid metabolizer, poor metabolizer. And what we do is we really enhance the toolbox of the physician, and we can really...
Enhancing the toolbox of the physician, yay, because I find out then that I can use this medicine. It'll work, but this one won't.
Exactly,
Yeah.
and it may empower you to pick a dose of the medicine that will work better for that patient, and you can personally tailor the medication to the individual, and that's really what we're aiming for with pharmacogenetics. Here in our genetics lab, we want to translate what we do at the bench to the bedside and ultimately help our patients.
So what drugs are we looking at mostly? I mean, psychiatric drugs mostly, right?
It can be psychiatric drugs.
Plavix, which is
Yeah.
a platelet inhibitor.
Yeah, so people, there's some very good data that just was released, which shows that individuals who do get pharmacogenetic testing after they've had a stent placed to determine whether or not they're good responders to Plavix, if they have the testing and are found out that they don't respond well to Plavix, they get put to a different drug. And data has shown that actually reduces the stent rethrombosis. So it's a really huge finding. And that kind of a change affects about 20% of people. So it's fairly common to see these
it's a
things.
huge thing right there, the risk of
So we
having
can
a stroke or
prevent
not.
that. So
Yeah.
we can prevent that by simple blood tests and
Or a
simple
heart attack.
pharmacogen.
Yeah.
Wow.
And the harms to pharmacogenetic testing are fairly.
Cost is about it, right?
Cost,
But we're
yeah.
down to $250 or something like that for screening. Other drugs, I mean, the psychogenic medications. I mean, people with antidepressants.
Antidepressants are a nice target because they go through a pathway in the liver that we can look at the genes that break down those medications. Pain medications.
But that's huge.
Yeah.
Certainly if you're a person who's in pain.
We want to make the patient comfortable after surgery. So if we can do that by looking at their genetics.
I'm all in favor of that. Okay. So what is the piece of genetics that makes twins alike or different? Research is the key to the answer.
The Avera Twin Registry began here back in May. That's our launch. However, the history of the Twin Registry goes back 8-9 years with our collaboration with the Free University in Amsterdam and the collaboration with a lady called Dr. Dorrit Boomsma. We've been doing twin research since then, and I think a natural progression from that was to start our own twin registry here in South Dakota and in the Midwest.
Avera looks to them as a model. They've been in existence for 30 years, and it's very successful.
If you look at the work that Dr. Boomsma and her team have done in the Netherlands and the impact that it has had to the genetic understanding of a disease, and then to think that she's a co-partner and that Avera is going to be starting something similar here in the United States, that's why you should be a part of it, is there's a very rich research opportunity right here at Holm.
We also know that the expression of the genome and epigenetics depends on environment. So it's also fantastic to have an opportunity to compare results across two cultures that are different, one in the Netherlands and one in South Dakota.
We have a lot of people come in who are twins and don't realize that their family can be involved. And it's really an exciting thing to be able to have all the siblings, all the parents, grandparents. It's important for research because genetically we can look at the linkage between them, genetically and environmentally.
I mean, those are things like what's the penetrance of disease states? Like if she were to get a breast cancer, should I be worried about it? But other things that come up with twins, like we're just fascinated by our children.
Yeah,
because they look very alike and they relate with each other as more like siblings. What's the piece of genetics that makes me and her and like our personalities are somewhat different, but we have so many things that are like we both like music, but we played separate instruments. Like how does that How does your genetics determine that pathway? And I think most twins really, really appreciate that and know that it's a special relationship. So that's the other piece is, you know, just whatever they find out from a research standpoint, if we can be a part of that just because of who we are, I think we're pretty excited about that.
This is actually something we can make a difference about, make a difference in health. We can personalize medicine based on the information we get, personalize determining whether you're at risk to have a certain disease or a certain disorder, act early on that prediction or risk.
I think I've also seen it really engender a tremendous sense of community among families with multiples, families of twins. There's been it sets you up in kind of a social network that I think you might not quite obtain or access in the same way without that. It becomes sort of a hub for all things related to living as a twin, as a dizygotic twin, or as a monozygotic identical twin.
In genetics, collaborations now is the norm. I mean, the model of competition has been to a large extent replaced by a collaborative model because each group on their own simply doesn't have the populations, the cohorts and the resources to achieve the next breakthrough. So we have to work together.
That was fascinating. You know, the data that I have learned from twin studies was when I was doing research on obesity. And in the Netherlands, they had identical twins that were split between families, raised in different environments, even though they were identical twins. And they found that it wasn't the environment they were raised in, but it was the genetics. They weighed as adults what their biologic parents weighed, not what their environmental parents weighed. Isn't that fascinating?
Yeah,
I mean, it tells us that despite our wishing that we could be something different, you know, thinner or heavier, I mean, we're going to be kind of what our parents tell us. It's more of the genetics than the environment.
It is fascinating what twin studies have contributed to the understanding of common complex diseases and where you can, I mean, like the heritability component of coronary artery disease. We know that because of twin studies. It's done a lot of fascinating things for the field of genetics.
Yes. And
well, twins are extremely valuable to disease studies and to the field of genetics. With twins, you know, we can really determine how much, like you said, is related to genetic factors and how much of a certain trait or disorder is related to the environmental factors. So for example, two people, one may have high blood pressure, one may have low blood pressure. What's causing that? Is it the genetics? Or is it the environment? Are we eating too much salt? Well, with twins, we can really start to answer that question. And it really lies in the fact that there's two types of twins. There's identical twins, which share 100% of their DNA, and there's fraternal twins that share 50% of their segregating DNA, and they're no different than brother or sister.
right.
But if we compare blood pressure between identical twins, and we compare the blood pressures between fraternal twins, if genetics is a large component of that, you would expect the identical twins to have equal, really similar blood pressure readings, whereas the dizygotic twins might not have as closely matching blood pressures. And if the correlations between the identical twins and the fraternal twins are similar, then we can say the environment is influencing it more than the genetics.
And we know that they both do, don't they? So we're actually looking for people who are twins or know of twins, that there's a number they can call, right? We can put that on the twin registry here. So we will give you a telephone number to look at, and well, there it is.
Yeah, and earlier this spring, we at Avera Institute for Human Genetics, we inaugurated our own twin register. So we currently have approximately a little over 400 individuals enrolled in our twin register.
Okay.
And of course, you know, as we saw, we collaborate with the Netherlands Twin Register too, and that gives us the unique opportunity to look at that environment, that environmental component, and how that influences certain traits and diseases. The environment in the Netherlands is significantly different than the environment here in South Dakota.
They talk different than
They
we
talk different, but they exercise different too.
Oh, they probably do, don't they?
Everyone's riding a bike and they eat different foods. They eat a lot of fish and herring and cheese. And here in the Midwest, we're a little bit more sedentary. We all have cars. We're all driving to work.
We still eat a lot of cheese.
Cheese is good, yeah.
Well, you know, it's not going to cost anybody to
No,
call
it's
that
no cost at all.
registry. You get involved. They don't, you
And you
know,
can contribute to science. And one other thing.
Knowledge.
Knowledge.
There it is.
And one of the things we can do, because when we collect a DNA sample, we look at the DNA, and one of the things that we can provide is what we call zygosity testing. So you may want to know if you're an identical twin with your co-twin, or you may want to know if you're a fraternal twin. The only way to really conclusively know is to look at the DNA. So that's something we provide all of the participants in the
now, there's a
research.
lot of, people say that there's a lot of difference between people. For example, we have a picture here of Wilt Chamberlain and another picture of a shorter person. And so they must be very different genetically, right? Or how different are they? Cassie, what's the genetic difference? A lot of difference between those two people?
Well, I mean, we actually do know a fair amount about the genetics of height. It's not my area of expertise, but you know, the majority of their genome is the same.
Just exactly the same.
But yeah, obviously there's a little variation there in the height.
Well,
bit.
in the color too, I think.
Yeah.
And their skin color and everything, except that they are genetically so close still. Isn't that amazing?
Yeah, we all share the vast majority of our genome with one another, and there are teeny tiny variations that probably don't mean much at all. That's what makes us unique.
There it is. We like that too, don't
So
we?
the first human genome was sequenced in 2002. And what that means is putting in order all 3 billion nucleotides that are in our DNA, the A's, the T's, the G's, and the C's. And one of the major take-home messages of that Human Genome Project was how similar we are. So if you would take your 3 billion A's, T's, G's, and C's and line them up and put mine right underneath them, 99.9% of them would be exactly the same. 0.1% would be different. And that 0.1% is what makes Wilt Chamberlain grow tall. What makes Willie Shoemaker, the jockey, a little bit shorter.
Yeah.
It's what gives me freckles. It's what gives me blue eyes.
You have blue eyes, I have brown eyes, but that otherwise we're just about the same, aren't we?
And unfortunately, that 0.1% is what puts us at risk for disease.
Yes.
It's what makes us metabolize drugs differently. And as geneticists, that's what we're interested in. We really want to examine that 0.1% and to study that.
You were talking earlier about the twins, and I was thinking about a comment that I'd heard about fecal testing and the whole thing about biotomes and the environmental things that are happening. I mean, we live in a world of infection. Microbes that surround us. There are more microbes on our body and in our body than there are cells of us, right? I mean, we are a slurry of the environment as well. So how important is that? That's an environmental thing, right? Not a genetic thing?
Both. So you're referring to the microbiome.
Yeah, that's what I meant.
Which really means all of the microorganisms in an environment. So we're talking bacteria, viruses, yeast, fungi, and we have 10 times more cells of those microbes than we have our own cells.
Gives me the heebie-jeebies.
And in human and disease research, we're interested in the microbiome that inhabits us as humans. 99% of them are in our gut. They're in our intestine.
Well, and that's that C. difficile overgrowth whole thing and antibiotic overuse and so on. Where would you go with that, Cassie? The C. difficile is something that we really
struggle with.
Fear.
Yeah, well, yeah, because it is very common. It's out there, but we have made people a lot more susceptible than probably we need to by using a lot of antibiotics, which kills our internal our internal biome that protects us.
Right.
So it's,
Yeah.
you know,
And
it's
there's a
risk.
whole new era of probiotics. So you hear
Yes.
of yogurts and pills where you can, actually ingest beneficial bacteria to help change that environment within our guts. So those bacteria in our gut, we have about 20,000 genes, but if you add up all the genes of those microbes and on us, we have about 3.3 million genes of those. And those all affect our appetite, they affect our mood. There's really preliminary evidence that those genes and the gene products from those microbes that inhabit us also affect our depression, with anxiety. So it's really naive to think that all of those microorganisms that are on us, and if you actually weighed them, it would be about 3 pounds, the size of
Oh, is that right?
the size of a human brain. That they have a physiological effect in our health and our disease.
Yeah.
And what we do is we can actually, you mentioned taking a fecal sample. So some of the participants in the Netherlands Twin Register will actually provide that to us. And we can take that fecal sample and we can look at the DNA of those microorganisms, and we can then get an idea of the number of microorganisms, the diversity of microorganisms. One of the thoughts is that the more diverse number of microorganisms, the more you have of the different classes, the more healthy you are. So
Diversity is health.
Yeah.
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 Well, they talk about diversity being healthy in the trees of a small community. If all you have is Dutch elm in that community, and Dutch elm disease comes in, the town is without trees. If you've got a mixture of all sorts of different kinds of trees, the Dutch elm may come in and wipe out one, but it's not going to be spreading that easily through the whole town. It'll maybe hit one of those, but a lot of Dutch elms won't even be affected because there's all these other trees that are around.
I would say that's a very
is that a good
analogy.
example?
Yeah, it is.
Well, that's the South Dakota deal, because Dutch elm disease wiped out many of the beautiful trees in South Dakota, Iowa, and Nebraska, and surrounding states. So they say that the more diverse we are, the better. That leads to where is the greatest diversity in the genetic pool of the world? Have you heard that discussion? They say in the middle of Africa is the most diversity.
Yeah.
If you go up to Northern European, we are so close that Dutch elm could wipe us out.
Yeah.
Any comment as
but, and that's been an area where some of our understanding of common disease is lacking. Many of the genome-wide association studies that we look at for things like heart disease and diabetes and hypertension really do focus on a Western European, Northern European population.
Right.
And we're just now starting to have more studies in African American populations, Hispanic populations, because obviously the genetic predispositions are going to be different among these people. We know that by prevalence of disease.
Yeah.
And so we have a lot to learn about the genetic composition of complex diseases in South Dakota are the minority populations, and the rest of the world is probably the majority of the population. So that's an area that's growing rapidly, I think, too.
We haven't said a word about epigenetics yet. I mean, to me, the new thing that just has blown me out of the water is to realize that the turning on and off of genes can be inherited. And that if I were a smoker, my grandchild, who never knew me, might be influenced by my smoking. Could you explain that a little bit, Cassie? Turn it to this guy.
Yeah.
Well, that epigenetics is a very, very hot area of genetics research, and you hit the nail on the head where we talk about gene activity. Turning genes on, turning genes off, and what that really means is all the cells in our human body have the same DNA, have the same 46.
Every single cell.
Every single
is
cell.
Same DNA.
But what makes a skin cell different from a nerve cell, different from a spleen cell, different from a liver cell, are what genes are turned on and what genes are turned off. And that's really controlled by epigenetics. And by epigenetics, we mean, epi, it means on top of. So epigenetics is gene activity changes that don't affect the sequence of the DNA and are inherited to successive generations. And that's typically accomplished by little marks that sit on top of the DNA and tell them to turn on or tell them to turn off. And those marks are placed there through environmental signals. So you mentioned smoking. It could be playing violin. If you play violin, you may have some methylation or these marks that change the DNA and influence neurotransmitters in your brain that
you can hear music better.
You may focus a little bit better. You may have more concentration. Imagine having a violin and being able to put the bow to that and to play a nice classical piece of music. That takes a lot of focus. That takes a lot of concentration. That can really model the brain and change it in a way that's beneficial. Alternatively, by smoking or by eating too much or by not exercising, you can also affect the gene activity changes.
Your children will inherit these things.
And there was actually a classical Dutch study where they went through a period of famine in the 1940s, and mothers who were pregnant, who didn't have a lot of food, their children actually then compared them to children that were born in times of plentiful food. The children that were born when there was ample food actually were more susceptible to heart disease and other types of disorders as well.
Even a little bit of starvation in those mothers benefited the children. Go figure.
And it's all through epigenetics.
It's amazing. In our world, we seem to be having more and more obesity. Mm-hmm. Cassie, do you think that has an epigenetic result? Of course it does.
Yeah, of course it does. I mean, we know that the epigenetics plays a role in probably most of our health. And, as we see our obesity rates increasing, it's kind of exceeded the stage of an epidemic. It's almost like the norm. People expect obesity. But yeah, that really truly will affect our children.
Yeah. So what you eat now doesn't only affect you.
Yes.
It also affects your children.
Wow.
So if that's not motivation for a healthier diet.
Let's get exercising, let's not eat so much, and let's play the violin. I mean, that's what I've learned from this show today.
Right.
And now for the answer to tonight's Prairie Doc quiz question. Who established the concept that evolution occurs as a result of natural selection and survival of the fittest? Was it Mendel, Darwin, or Lamarck? And the answer is Darwin. We'll be back right after this.
All around town, from stores to playgrounds, babies are on the move. And there are diseases that are on the move too. And some of these spread easily. To best protect him from 14 serious diseases by the time he turns 2 years old, vaccinate him according to the recommended schedule, so he can go on about his business and you can have peace of mind. For more reasons to vaccinate, talk to your child's doctor or go to cdc.gov/vaccines.
When did we first understand what directs the shape of our nose, foot size, personality, and chance of surviving in a tough world? There's an interesting history about how we learned that our ancestral genes and environment are responsible. Hippocrates and Aristotle stated that heredity material gathers from throughout parents' bodies and mixes during the sexual act. About the same time, ancient scholars from India added 2 more factors of heredity. They added the soul, which enters the baby from some undefined place at some undefined time, and the diet of the mother. In the mid-1700s, Carl Linnaeus, a brilliant Swedish botanist physician, believed that both shared parental fluids and environmental factors influenced inheritance. Late in the 1700s, Jean-Baptiste Lamarck proposed that animals can acquire characteristics influenced by their environment and pass that on directly. His famous example was the long neck of the giraffe that he thought resulted from parents stretching to reach leaves. He thought this characteristic was then directly passed on to offspring. In the mid-1800s, genetic understanding was greatly advanced by Friar Gregor Mendel, who worked mostly on pea plants. His influence is still taught in biology classes. And about the same time, English naturalist Charles Darwin put together observations of animals and plants from a 5-year around-the-world sailing expedition on the HMS Beagle. His revolutionary and famous work came out in 1859 entitled On the Origin of Species. Darwin defined evolution as the result of natural selection, acting on random mutations. Put it another way, each species improves only through the survival of the fittest of each subsequent offspring. For example, the weakest wolf pups of the litter don't make it, but the strongest are best able to reproduce with a mate and have offspring, thus passing on the advantage through selection. Darwin's natural selection theory clearly dismissed Lamarck's giraffe environmental example. Scientists felt that with Darwin, they finally had the answer to the puzzle. But controversy followed Darwin like a pack of wild dogs. Darwin was opposed by many religious leaders since God seemed removed from that process. Darwin, a very religious man, believed the hand of God worked through natural selection, which some might define as Darwin's theory of intelligent design. That did not and still doesn't satisfy certain believers, but that's a discussion for another time. Most recently, we've discovered that environmental factors like starvation, smoking, or stress can activate or suppress genetic codes, and those changes can be passed on, reviving Lamarck's ideas of environmental influence. I don't have to stick my neck out too far to say that there is great cause for excitement in the emerging field of genetics and epigenetics. Well, we have a great big thank you to our guests tonight, Cassie Hajek and Erick Ehli. This has been a fascinating experience and a discussion. Thank you for coming here, for your time spent joining us tonight for this discussion on genetics. And we would remind you too often that the flu is still here and it's going to get worse. Get your flu vaccine now to reduce your chances of catching the flu bug this season. Well, that does it for tonight. From all of us here at On Call with the Prairie Doc, until next time, stay healthy out there, people.
Can't get enough of the Prairie Doc? Then visit our webpage and other online programming. You'll find extended interviews with guests and more information on important medical subjects. Visit prairiedoc.org or follow us on Facebook. The best way to stay healthy out there.
Major funding for On Call with the Prairie Doc has been provided by
Avera is a proud sponsor of On Call on South Dakota Public Broadcasting.
LARSON Manufacturing is proud to support On Call Television as it continues to open doors for important medical information.
And by the South Dakota Foundation for Medical Care, the Medicare Quality Improvement Organization for South Dakota,
and with the ongoing support of these individuals and institutions.
Brookings Health System,
Ophthalmology Limited,
South Dakota State Medical Association,
Avera Heart Hospital,
Dakota Allergy and Asthma,
Fishback Financial Corporation,
Vance Thompson Vision,
Aberdeen Asthma and Allergy,
Black Hills Medical Society,
DAKOTA Bank,
Orthopedic Institute,
Physicians Care Sanford Clinic Community Service Committee,
Regional Health,
and Swiftel Communications.